All right, so welcome to the lecture on genetic engineering. Something to keep in mind, this is not going to sync perfectly with what's in your textbook. Reason being, I'm trying to introduce more relevant content and newer updates and help you guys make connections into life experiences in which genetic engineering has become a part of. So don't panic if you're reading this or watching the lecture and you're using the book and it doesn't match exactly. That's okay. It's not going to line up perfectly. But genetic engineering is an incredibly changing field of science. There are new discoveries happening all the time. Things we talk about during this section of the course, during this semester, are going to be different in a year, 2 years, 10 years. Imagine where we'll be in 20 years from now with genetic engineering. I mean, if you're old enough, think about 20 years ago, where were we at with this? It was barely even on the table, and now it's just ingrained into our lives and permeates nearly everything we do on a daily basis. So that is a big thing to keep in mind when we're discussing genetic engineering, is that it is a rapidly changing field of science, and a lot of those changes are because of technology and how technology is working and advancing. So what I want you guys all to do during this chapter is start to formulate your perspective and opinions on genetic engineering. Formulate it based upon knowledge and information and facts, things that you can use to back up your opinion and support your opinion. Do you think we should do this? Should we continue to do this? Should we stop this? Should we monitor this? How do we regulate this? How do we control this, etc.? Come up with opinions and support them. Back them up with the fact and the information that we're going to discuss through these lectures and other things you can find out there that actually have scientific basis to them. Not, there was this thing I saw on Facebook, or, oh, I heard this in the news. Where is the evidence that supports the information you're using to create your opinion? So definitely an important thing to do when we discuss a topic that sometimes can be very controversial, like genetic engineering. All right, so let's jump into it. It's a fun topic, it's exciting, and I want you guys to have a better understanding of it. So the basic premises of genetic engineering is tweaking, altering, changing the DNA of an organism. There's a lot of ways to do this. I mean, humans have been doing this for 12 to 15,000 years. We do it through a thing, or we have been doing it through a thing called selective breeding. I'm going to take that animal and breed those two dogs, breed those two kittens or cats, breed those corn plants, etc. That is manipulating the genetics of the offspring. To some level, that is a form of genetic engineering. We're now doing it in a far more advanced way, a way that can change things much faster, sometimes less than a generation. All of a sudden you have a new gene inserted into a species. What does that do to that species? So we're going to talk a little bit about it, some of the technology behind it. All right, so here's one technology. It's called recombinant DNA technology. And when this is being used, what's happening is you're actually mixing the DNA of different species together, usually taking a gene from one species and putting it into a second species. So in order to do this, you need certain tools. The first tool is called a restriction enzyme. This is an enzyme that is used to cut the DNA at a specific location. I want to cut the DNA at chromosome 7, and I want to cut the base, starting at 287,000 letters long. I want to start cutting there and pull out a section that is 8,000 letters long, 8,000 nucleotides long. That's what restriction enzymes do. In a sense, they're high-tech microscopic scissors that cut the DNA at a certain location. Once you have that DNA, you need to be able to get it into something else. And that's where this thing called a vector comes into the picture. So a vector is an organism or structure, substance, etc., that will insert the DNA into the host. Quite often, viruses can be used for this. So if you think about a virus, and we often, oh, I hate viruses, I got the flu, I have this viral infection, that one. The virus itself is not the problem. It's the genetic information the virus carries that gets into your cell and causes your cell to do what it's going to do. So imagine putting a good piece of DNA that's going to heal a genetic disorder in a person. You put that into a virus. You want that virus then to infect that host. Put the new DNA into the individual who needs it, and now they're infected, but infected in a good way. So the host is the organism that receives the new DNA or the new gene. Okay, so maybe I'm the host in this genetic engineering experiment, and I need a new piece of DNA that's going to fix cystic fibrosis. Because my DNA, when it comes time to make the protein in the cell membrane, it's not correct. And that's what's causing cystic fibrosis. Or we talked about diseases like Huntington's disease or Tay-Sachs or these other genetic disorders. What if it's just cancer? My gene that is supposed to stop tumor growth isn't working. Can I get a good tumor suppressor gene into my body? Well, maybe I can do that with recombinant DNA technology. But I'm the individual receiving the DNA, so I'm the host. The donor is the organism, or it could be an individual, that donates. Make sure I spell this right. Donates the new DNA or the gene. So where do we go to get the gene? Depends upon the gene we're looking for. Find that donor species, organism, individual, take it out of them, put it in a vector, insert it into the new host. Okay, so that's the very broad overview of biotechnology with a recombinant DNA example. There are lots of ways that this is being done, lots of different methods and options. It just depends upon what is the end goal. What are we ultimately trying to do for the organism, change the species, cure a disease, etc. So another type of technology associated with biotechnology is a thing called PCR cycling. PCR cycling has become very routine biotechnology practice. It's where we actually copy the DNA in vitro. In vitro means basically in a test tube, petri dish, whatever you want to use. It's not naturally copying in nature, it's controlled copying in a laboratory environment. So here's the example. Let me go to the picture. Alright, if anybody saw the movie, Robert Downey Jr., Robert Duvall, boom, car accident. There's blood on the car from a victim. Somebody got injured in this accident. Driver drove off. Police pulled the vehicle over. Okay, you have blood on your car. We're going to take that as a DNA sample. Does that match the person that we found on the road that was injured, that got hit, a hit and run accident? So maybe you only have a small amount of blood off the hood of that car because the rain washed it off, or in some cases the driver of the vehicle washed the car, whatever the case may be. But you only need a fragment of DNA. You need one cell, really. One cell that contains DNA, and then you can amplify the DNA. So you take that one single cell, extract the DNA from it. Now you have DNA in your hands. You take that DNA and you do the PCR cycling. So you start here. In a controlled laboratory setting, you rip apart the DNA. So you need a DNA helicase. Heat it up. Strand opens up. You add primers to the exposed strand. Right there, right there. And now you put new nucleotides in the solution. and then you get the DNA polymerase that comes over and attaches the new nucleotides to the original strand there. And it basically is replicating your DNA in a controlled laboratory setting. So you do that PCR cycling once, you have 2 copies. Okay, we need more than 2 copies to sequence the DNA. Let's do it again. Another cycle. Now you're up to, so here's your 3rd cycle. You're up to 4 copies. You do it again. Here's your 4th cycle. You're up to 8 copies. So think about 2, 4, 8, 16, 32, 64, 128. Boom, 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 boom. How fast can you get a lot of DNA through PCR cycling? Now once you have all that DNA, now you can sequence it. Now you can look at the genes in that DNA to see if they match to the victim. Do they fit that victim? Okay, so here was another really cool experiment or example of how biotechnology gave us knowledge about something that happened hundreds of thousands of years ago. Back in 1991, hikers in the Swiss Alps found this body. They thought, okay, people die hiking in the Alps, killed in an avalanche. They call the police. They bring the body out. They start doing an autopsy, and they start noticing stuff's not right. Things aren't what we call modern. Look at the weapons. Look at the tools laying on that green cloth. Those aren't modern. Those aren't even 100 or 200 years ago, or 200 years old, or 300. They've been around a lot longer. So they did DNA analysis on this guy. This corpse is known as the Iceman, because he was found in the ice. And they went down into his digestive system, and they pulled out contents out of his stomach and his digestive system, small intestine, large intestine. And they discovered, well, he has the DNA of bread in his stomach, wheat DNA in his digestive system. He has DNA from certain animals in his stomach because he had consumed meat. And he had DNA from certain greens, different plants, in his digestive system. So this individual died over 5,300 years ago. It appears he was killed. Still piecing together how that happened or why. But forensic scientists used DNA sampling, PCR cycling, all these different techniques to figure out what this man ate. And they knew he died in the springtime because the plants in his stomach only grow in the spring in the Swiss Alps. So the assumption, if you have fresh greens in your stomach, you died during the spring when those plants were growing. You didn't die in the middle of January when there is no plant life in the Swiss Alps because it's all in dormancy. So it helped piece together a puzzle of this man's life when he died over 5,000 years ago. And that's the power of biotechnology today. That's how we can use it for some really incredible discoveries. All right, so keep in mind, this is a constantly changing topic, field of study, etc., because technology is constantly improving our ability to make advances in biotechnology, understanding these connections between species on Earth. So in our next lecture, we're going to jump into Jurassic Park. Take a look at this and look at how did Jurassic Park, movie that came out back in the first one, came out back in the 90s, how did that actually use DNA for the movie? How did Hollywood play off of DNA and genetic engineering and biotechnology to create this multi-million-dollar franchise set of movies? And is Jurassic Park realistic? Is it possible to do what they talk about doing? There's lots of things that we'll discuss when we get into this, but in the next lecture I'm going to show you a little clip from the movie. We'll talk a bit about it, and we'll see how biotechnology is used by Hollywood as well as by science.